All right, in chapter 8, we start introducing metabolism. So chapter 9 is going to be cellular respiration. Chapter 10 is photosynthesis, some of the most complicated metabolic processes out there. So in this chapter, my goal is really to introduce these concepts, to help you understand some of the ways that matter and energy interact so that when we get to things like cellular respiration, it won't be quite as confusing. So the thing to keep in mind, right, when we talk about metabolism, it's not like, oh, who has a fast metabolism and is skinny, right? Or who has slow metabolism. Really, like, this is like super nerdy. It is the sum of all chemical reactions in the body. So the thing to keep in mind, right, is the cell that we've been talking about. It's just this miniature chemical factory where thousands of reactions are occurring every second, right? So cellular respiration, where we're heading next, is how we extract energy stored in sugar and other fuels. So cells apply that energy that they get from their food to perform work. Some organisms even convert energy to light, as in bioluminescence, right? So there's a huge variety of different metabolic reactions and processes out there. So, okay, here's a key thing to understand, that a lot of these metabolic processes are set up in what are called pathways. So if metabolism is the sum of all the organism's chemical reactions, right, a metabolic pathway is how we organize those reactions. What we see here, right, is we have a starting molecule and we have a product that we're trying to make, right? So we'll talk about substrates and products. This metabolic pathway, each of these reactions, right? So when you see an arrow, they're trying to show that something is changing, there's a reaction. Each of these is catalyzed by a very specific enzyme. And so the enzymes that different cells, different organisms make are going to dictate what these pathways look like. And so the way I think of it is kind of like, here's a map of Steamboat. So we're up here on campus and maybe you wanted to go get a sandwich. And so you're going to have to go through a series of different, intersections, which are kind of, the different steps in a metabolic pathway. Here's the real metabolic pathways, right? We actually have this chart hanging on the wall in the lab. This is trying to lay out the metabolic pathways that are taking place, I believe, in the human body. Down here, I can actually recognize we've got some cellular respiration going on down here. So I like this because it shows there's a lot more going on. Maybe you can think about it like a road map, right? If I'm at point A and I want to get to point B, what do I have to do? There's a series of steps. And in traffic, you'd have things like traffic lights, stop signs that are maybe dictating like when you can start and go. And then in life, the analogy continues, it's enzymes, right, that are determining kind of how these metabolic pathways play out. Okay, so in metabolism, the sum of all metabolic reactions, I did want to point out we definitely split metabolism into two main categories: anabolic reactions and catabolic reactions. So to help you remember this, I've got a couple of images here. So anabolic, I think anabolic steroids. So Arnold Schwarzenegger took anabolic steroids when he was, Mr. Universe or whatever. An anabolic reaction takes simple molecules and puts them together to make something more complex. So for example, anabolic steroids helped Arnold take individual amino acids and build more protein. That's why his muscles are so huge, right? Anabolic pathways require energy. So we say they're endergonic. They require energy to build these more complex molecules. Now, catabolic reactions are taking complex molecules and shredding them apart into smaller, this is just to help you remember it, right? Breaking those complex molecules into simpler compounds. Catabolic reactions release energy. So the classic catabolic reaction we're going to look at is how you take glucose, C6H12O6, right, complex molecule, and break it down into nothing more than CO2 and water, carbon dioxide and water, right? In that process, we release energy. And so what we're going to see in metabolism as a whole is it's really that Catabolic reactions and their release of energy then provide the energy for anabolic reactions to take place, right? And the link between those two is going to be ATP, as we'll see. So there's a lot having to do with how energy moves through organisms in order to understand metabolism. So you can get into a little bit of physics in this chapter, right, where we talk about potential energy and kinetic energy, right? So this diver up here on the platform, I should say, so energy is the capacity to cause change. It comes in different forms. Kinetic energy is energy associated with motion. There's thermal energy, right? How the kinetic energy of molecules in this, say in this water here, they're moving around. That's the thermal energy. Heat is actually thermal energy being transferred between objects. Potential energy, that's where I was going to go with this guy. Potential energy is energy that matter possesses because of its location or structure. Chemical energy is a form of potential energy that we'll deal with a lot. So in this example, potential energy, right? By climbing up these ladders, you're basically storing energy. You have the ability to release that energy, right? This guy's in motion, so that kinetic energy is being released. And you know there's energy when he hits the water, and this kind of looks like it's going to be painful, right? You're going to hear the smack. Sound is that energy being released, right? He'll feel the heat, right? The friction of his body entering the water is going to produce heat. That is energy as well. So it's important to understand potential energy, right? The person standing on this platform has more potential energy, right? The ability to do work than this person just down here floating in the water, right? And climbing those ladders, right, is taking kinetic energy and storing it in this form of potential energy. So for some people, this helps. To me, I think, what we're going to talk about a lot is chemical energy. So in food, in glucose, between those carbon, in the carbon bonds, right, in glucose, there is energy stored there, right? It's potential energy. And so when your body breaks those bonds, it can release that energy and then do work, right? You can do your homework. You can go for a run, those sorts of things, right? So it's a lot of conversion back and forth between potential and kinetic energy. So, the bear. Did you guys follow the fat bear contest? Right? They keep track of the grizzly bears. I forget where that is. Is it in Yellowstone maybe? And there's a contest. Anyway, check it out. So since we're talking about energy, it is important. It should help you understand by understanding these two laws of thermodynamics. So thermodynamics is just a fancy way to say you're studying energy transformations. You'll make your parents proud if you tell them you went and learned about some thermodynamics. So we've really got two different kinds of systems. Isolated systems, so if you have liquid, say, in a thermos, it's unable to exchange energy or matter with its surroundings. We'd call that an isolated system. Organisms are open systems. Energy and matter are being transformed between the system and the surroundings. And so if you think about our open system, what we see in these two laws of thermodynamics, right, is the first law basically says energy can't be created or destroyed. Energy can't be created or destroyed. That's the first law of thermodynamics. It can be transferred. So this bear is eating a fish. There is chemical energy in that fish, and he's going to transfer that into himself. Some of that energy he might convert to kinetic energy to run. Some of that energy is being used to heat the bear. Some of that energy is going to get turned into triglycerides to be stored on the bear for later, right? So he might turn the fish, which is potential energy, into body fat, which is another form of potential energy, right? So we can just keep transferring this around. But energy is not created or destroyed. The thing that the second law of thermodynamics says, though, is every time you transfer energy, right, when it moves between the fish and the bear, and right, every time you transfer energy, some of it becomes less useful, right? Some of it, notice the heat, right, is dissipating off into the environment. So during every energy transfer, some energy is unusable, often lost as heat. We'll see that. And the second law of thermodynamics says, yeah, every time you transfer energy, you lose some of it. Some of it goes towards increasing the entropy of the universe, right? Entropy is a fancy way to say disorder or randomness, okay? And so it's interesting. So basically, oh, this is so into physics. Everything in the universe is moving towards disorder, right? Life is really unique in that it displays order. Everything else, think about like your car, right? It's rusting out. It's going back to chaos. Your room, right? You organize it and then it all wants to go away. Even if you look at the universe, right? The universe is expanding. What was like condensed and compact is moving towards disorder, towards entropy, spreading out. So life ends up being this like weird pocket in the universe, right? The only thing that isn't like moving more towards disorder and entropy is living organisms, right? And we do that by eating, right? We use metabolic processes. We take the energy from food to order ourselves, to fight that trend towards entropy. Now, of course, once you die, that doesn't work anymore and you return to entropy. But while you are living, that is such a cool thing about organisms, right? Life displays order. Okay, you get the joke. So, right? So again, as we think about our bear here, right? When we start to look at all these metabolic processes, these chemical reactions that are happening, right? There are some processes that are spontaneous. They occur without energy input. Now, that doesn't mean they're necessarily fast, right? They can happen quickly or they can happen slowly. But to occur spontaneously, it has to increase the entropy of the universe, right? Things that decrease the entropy of the universe, i.e. anabolic reactions, are non-spontaneous. They have to have energy provided in order for that to occur. So organisms are creating these ordered structures from less organized forms, right? We're taking amino acids and we're building proteins. Okay, I think that's what I want to say. Go back through there. Okay. Excuse me, talking too much. Okay, so then in the chapter, they get into what's called Gibbs free energy. And I don't want you to get lost in here, right? There's math. It looks really complicated. I thought this picture did a really nice job of summarizing the relationship between free energy, stability, work capacity, and spontaneous change, right? So in a living system, right, and we said we're open systems, the free energy is the energy that can do work, okay? And they represent that with this delta G, right? Delta means change. So the change in free energy during a process, how do I want to explain this? Let's look at the picture here. So here's our diver, right? There we go. Here's our diver. He has more free energy and it is less stable, right? He's going to fall and has greater work capacity. Once he's in the water, right, he's going to have less free energy, right? He's more stable, but there's less capacity to do work. Okay. So the diver is hopefully an image that can help you understand, right? So the release of energy when he comes off this diving board is a spontaneous change, right? Because the free energy of the system decreases, right? More free energy, less free energy. The system becomes more stable. And that's what molecules want to do, right? Is to become more stable. Now, when he dives, right, that released free energy can be harnessed to do work. We'll hear the sound of his splash, right, and feel the heat. But in chemical reactions, we might be able to harness that energy. And so a lot of times what we're going to see is how molecules do this. So when you see molecules kind of clustered together, we were talking in the last chapter about diffusion, right? They want to spread out because staying together in this organized fashion, right? We have more free energy, but we're less stable. This is not where we want to be. We want to be at equilibrium, right? So this is going to be a spontaneous change. These molecules want to spread out. If we set this upright, the movement from that high free energy to low free energy, we can harness that to do work. We are actually going to see that in cellular respiration. We harness the diffusion of some hydrogen ions in order to do work. And maybe this will make more sense when we get there. Same thing, thinking about like a complex molecule like glucose becoming carbon dioxide, and water. Realize that the glucose has more free energy. It's going to be more stable once it's broken up. So this reaction wants to proceed. Now, glucose doesn't quickly go to CO2 and water just left to its own devices. We're going to have enzymes that encourage that reaction, but it is a spontaneous reaction and it's going to release energy that, again, we can harness to do work. Again, I know this gets kind of complicated. It's into the physics here for sure. Let's see if this helps. So if we have a reaction, so here's our arrow, this is the reaction. The reactants are on the left side of the arrow. So let's say it's glucose, right? When glucose breaks down to carbon dioxide and water, those products are more stable. They have less free energy. And so sure enough, we see this is the amount of energy that can be released in this example from glucose. This is an exergonic reaction, one that releases energy, one that is spontaneous, right? And that's what we're going to see out of our catabolic reactions. This lower panel is showing an endergonic reaction, one that requires energy. It's not going to be spontaneous. We're actually going to have to provide that energy. And so this is what it would look like if a plant wants to take carbon dioxide and water, two simple molecules, and build a product like glucose, right? That is going to take some energy, right? This much energy. And in our case, energy is provided by photosynthesis, or to photosynthesis from the sun, right? The energy is coming from the sun, and we can make this reaction happen. Okay, so those are endergonic reactions. This doesn't even matter. In an isolated system, you can hit an equilibrium, and then you wouldn't be able to do work. In life, we're always dealing with these open systems, and so even if you look at the cellular level, right, cells are open systems and they're experiencing a constant flow of materials in and out. So one of the features that kind of defines life is that metabolism is never at equilibrium, okay? This kind of, they're trying to show, hey, multi-steps, we can release energy every time we come up with a way to put a product at a lower, a more stable end result. This is very similar to what happens in your cells, where we release energy in a series of reactions. So the energy in a cell is going to be in the form of ATP. And so we talked about ATP a little bit with our macromolecules, but super important to understand. ATP is a nucleic acid, right? Notice here we have ribose and adenine as our nitrogenous base, right? Ribose is our sugar. But instead of a phosphate, right, now we have three, right? So think about like the backbone of your DNA, right? You had a sugar phosphate backbone and then a nitrogenous base. In this case, we'll have three phosphates. So the way energy is stored in ATP, again, is in the bonds. So we take adenosine diphosphate, right? If we have another phosphate and energy, we can shove that third phosphate on and make ATP. The energy is then stored in ATP, and all we have to do to get that energy back out is add a little bit of water, right? It's a hydrolysis reaction. The phosphate pops off and the energy is released. And so that is what cells are going to do. They're going to use ATP to do the work that they want. So cells are doing things like chemical work, right? They're pushing the endergonic reactions that they want to happen, the anabolic reactions that they want to have happen. They do transport work, right? We were seeing the sodium potassium pump. It's something like 30% of your energy goes to running sodium potassium pumps. Cells might want to do mechanical work, so like muscle cells actually contracting, and we need ATP for all of these things. ATP. Okay. Okay, this image is way too complicated, but basically what they're saying is that if you wanted to put glutamic acid and ammonia together to make glutamine, right, we'd need energy to make this anabolic reaction happen. This is not going to happen spontaneously. And so here we see the same reaction, but we're going to add ATP. Right? And that makes the reaction go. So notice putting the two molecules together that we wanted to requires energy. The ATP, when it hydrolyzes, it's going to release the energy, right? And that is what allows this anabolic reaction to take place. So overall, if you look at the numbers, overall it's exergonic, right? And that is going to increase the entropy of the universe, and so this reaction can take place. So the details of this get a little complicated, but I want you to think through that when we want an anabolic reaction to proceed, we're going to need to use energy. Okay, and then, so here's our, this would be like a sodium potassium pump again, right? We can use ATP to change the shape of that protein. Here we see a motor protein walking along the cytoskeleton, right, dragging something with it, and we might use ATP to do that. If you look at the details of it, again, it is a hydrolysis reaction. When you have ATP and you add water, that is going to split the phosphate off of ATP and go back to ADP. The energy that gets released can be used for any of those cellular processes that we were just talking about, right? So what we're going to look at more next week is how energy from catabolic reactions, things like cellular respiration, build ATP, so that we can have that in the cell to drive any of these cellular processes that we want. Oh, okay. So a little bit about enzymes. So, right, all of these reactions we said in a metabolic pathway are going to be kind of harnessed and run and directed by enzymes. So now we get a little bit to talk about enzymes here. And so we've talked about this reaction before where you can take sucrose, a disaccharide, and use hydrolysis to split it into glucose and fructose, right? This is one of the first things we talked about in this lab. Or, right, you could do this dehydration and put them together. So in this reaction, right, what we're pointing out is that this enzyme, so when the reaction happens, and that's the arrow here, we are going to use an enzyme. If you see this ASE ending on a word, ACE, it's an enzyme. This enzyme is just a catalytic protein. A catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction itself. So this enzyme is just encouraging this reaction to take place. And the way it kind of encourages this, right? Honestly, think of it as like enzymes catalyze reactions or enzymes encourage reactions, but this is truly what's going on. So if you have, these reactants and it looks like, yeah, we're going to switch. This is a little confusing. So we're going to change our bonds here, right? We want to get to A and C and B and D. If we do that, we are going to be more stable, right? We are actually going to have a release of energy. But notice what happens. So we have to break these bonds and form new ones. And that's true of every chemical reaction between different molecules, right? We're breaking bonds and forming bonds. The initial energy that you need to start this reaction is shown right here. So this just doesn't happen. We actually need this activation energy. So activation energy, you can use heat right? You can warm things up enough that these molecules are moving more quickly and things happen. What we're going to find is in living organism, heat denatures enzymes, and so that's going to be problematic. So if we can't use actual like heat energy to encourage these molecules to break bonds, allowing them to reform more stable bonds, what we're going to do in living organisms anyway is to use enzymes. So here's that same graph, right? We would have had to put all of this energy in, right? Activation energy in to get the ball rolling here. With an enzyme, that energy need is lower. So when we say enzymes catalyze reactions, they're really lowering the activation energy, right? And so this reaction, notice it started at a higher energy and went to a lower energy. This is an exergonic reaction. This is spontaneous, right? They just are able to speed up a reaction that would have occurred anyway. Okay, so enzymes are going to help us out a ton. So an enzyme is made out of protein. And a couple things that are important to understand here about enzymes, right, is they have an area called an active site. This is where the substrate, the molecule that you want to change, needs to bind. And this is a very precise mechanism. So a lot of times people talk about like a lock and key mechanism. The substrate, the molecule that you want to have the enzyme work on, has to fit perfectly in this active site. One of the things an enzyme can do then, right, is by bringing molecules into that active site, maybe it just brings them in close enough contact to kind of hasten reactions that would occur anyway. Let's take a peek. So notice in this, so we're putting something together here. Okay, so we have two substrates, and when they both fit into the enzyme together, right, notice what close proximity they're in. So in an enzymatic reaction, one that's driven by enzymes, the substrate has to bind into that active site. Enzymes tend to work really quickly, and they don't get consumed by the reaction, right? So this big purple blob here is gonna come out in its original form, ready to grab the next substrates and keep going. So basically they're getting recycled. So you don't need a ton of enzyme, like high enzyme levels, but they can still get a lot of metabolic processes done because they do get used repeatedly. So in this reaction, right, notice the fact that the active site, the proximity of these two molecules is what's going to lower the activation energy. And now we're able to make this conversion. And this is kind of hard. It's just like different colored blobs, but they're trying to show, right, that being that close together in the active site can really lower the activation energy and help this reaction to proceed. Once the products are released, notice, right, our active site is the same, our enzyme is the same, and we're going to keep coming. So the active site really, it can lower an activation energy by doing things like orienting the substrates correctly. That's kind of what this picture showed. It can help covalent bonds form, etc. Okay, a couple other things that are super important about enzymes is like the rate of the reaction, right, that an enzyme is catalyzing. It can be sped up if you keep upping the number of substrates. So we'll talk about this some more, but like if you keep increasing the substrate, you can get more product, and that kind of makes intuitive sense. But at some point, the enzyme does become the limiting factor. When all of those active sites are filled, right, we'd say that the enzyme is saturated. It can't move any faster. So then the reaction rate can only get sped up if you add more enzyme. Keep in mind too that since enzymes are proteins, their activity can be affected by the general environment. So things like temperature, pH, different chemicals. So I thought this was kind of fun, right? We see that, I just went away. We see that enzymes have an optimal temperature that they work at, right? We just said that they can be influenced by this. And so human enzymes work best at 37 degrees Celsius, which is body temperature, like 98.6. But there are some bacteria that live in hot springs where their optimal temperature is all the way up here at like 75 degrees Celsius. We would full on be dead, absolutely dead at 60, even at 45 Celsius, we're pretty much done, right? And so pretty interesting that we can have enzymes that work in such different environments. Same thing, pH can have a major effect here. So your stomach is extremely acidic, right? It has a low pH down around 2, and that is where the enzyme pepsin, which helps break down proteins, works super effectively. Trypsin, another proteolytic enzyme, works in the intestines, and it only works if the pH is much more alkaline. And so what we see happen is as the contents of the stomach with a very low acidity enter the small intestine, we actually dump a bunch of buffers that the pancreas makes to try to neutralize the pH and make it appropriate for the intestinal enzymes, right? And so those kind of factors can make a big difference on how enzymes function. I pulled this picture from another book just because I did want to point out that sometimes in order for an enzyme to work properly, it actually needs either a cofactor or a coenzyme. Cofactors are minerals, coenzymes are vitamins. And basically what we see happen here is the addition of that cofactor coenzyme is actually going to slightly change the shape of the active site, right? And so these enzymes won't work properly unless they have the vitamin or mineral they need. This is actually why a lot of vitamins and minerals are important in your diet, is to make sure that the enzymes can function properly. Okay, last few things. So the way enzymes then are regulated, right, turned on and off, notice what happens here. So this is the substrate. It fits perfectly, lock and key mechanism here in the active site of the enzyme, and we would expect a reaction to take place. But there are things that can make this not work. So competitive inhibition, we have an inhibitor that fits in the active site, basically blocking the active site. That is going to stop this enzyme from taking the normal substrate and making what you wanted. So you can actually use this as a drug intervention, right? If you can come up with a competitive inhibitor, this enzyme can't do its job. So for example, there's an enzyme in your lung called ACE that is important in changing some proteins that regulate blood pressure. If you take an ACE inhibitor, it's really just a competitive inhibitor that's blocking the binding site of this enzyme and making sure that you can't release these products that would actually up blood pressure. So ACE inhibitors help you keep your blood pressure down. You can also have non-competitive inhibitors, where here we have a different molecule and it's binding on a totally different side of the enzyme, right? It's not blocking the active site, but notice it does change the shape of the active site, because again, these are just proteins. And so anytime you add something new to a protein, the whole protein can change shape, right? And so non-competitive inhibitors can also keep enzymes from working and therefore shut down metabolic pathways. We use this some in the way we've produced antibiotics and pesticides, different toxins, etc. Okay, so that's just kind of your close-up of a protein. Okay, so this idea of allosteric regulation, basically what we're looking at here is our ability to kind of, for the cells to kind of turn on and off enzymes. We talked previously about gene regulation, like in the operon, how a cell can basically turn on and off enzyme production by gene regulation, right? By turning on and off the genes that encode for the production of certain enzymes. Allosteric regulation is when a regulatory molecule binds to the enzyme and affects its protein. And so what we're looking at here is quite often this picture is really kind of confusing. Quite often what happens is the enzyme itself the product that it's making can often bind back to the enzyme and slow its production. So as you start to build up high levels of the product, it's going to bind to the enzyme and change the active site, essentially slowing it down. Oh yeah, here's a great example, right? So here we have our substrate, an enzyme is going to be converting it, and they saved you the trouble, right? We're going through lots of different intermediate forms, but here's our end product. Notice that when enough of that end product builds up, it binds back to the same enzyme, and now we have changed the shape of the active site. Okay, and so this is another way just to like regulate how much how much work enzymes can do. So gene regulation is great. Decide whether you're making the enzyme or not, but then we also have this allosteric regulation. And this feedback inhibition is a form of negative feedback, super important in maintaining metabolic pathways in the cell. One way to kind of also keep things organized is to keep your enzymes in certain pockets of the cell. And so we'll see that if you go back to that huge chart of metabolic pathways, all of those things are not happening throughout the cell. Sometimes we use organelles to kind of give us a little bit of structure, a little bit of organization. So for example, here we're showing the mitochondrion, and that's where our enzymes for cellular respiration are going to be located. They're not kind of spread out willy-nilly. They're packaged here within an organelle. And this is just a fun picture of penguins jumping into the water. So I hope that helps. Chapter 8 is complicated, but I will say this. If you don't get some of these basics down, when we move on into the really complicated stuff like cellular respiration, we're just going to be lost. So do take the time to kind of think about this some more. Be encouraged if you're feeling like, man, this is super complicated. This is general college biology. Every class in the nation is doing this. There's a ton of resources between your textbook, Khan Academy, etc. So find some more videos. If the way I'm explaining things doesn't make sense, go seek out those resources that are going to help you. And we'll spend some class time answering questions and going over things as well. All right, have a great day.